Test, test. Okay. And one for your colleague. Thank you. Yeah, seminar. The two. Nice job in your lab book. Good job. Well done. Emily, can you hear me all right?
Yep, I can hear you okay.
Okay, cool. We're going to get started here. So,
Thank you.
okay, folks, time to get started today. I thought this news article was quite apropos given our situation today and given today's seminar. By the way, we've had very few people sign to come to the seminar. If you want to come, don't bother signing up, just come. It's in White Auditorium, 10 o'clock, and yeah, just show up. No need to sign up. Otherwise, Tom and I will be talking to not an empty room. Chuck White will be there. He always comes. Why we will never eradicate the coronavirus. We're going to have to learn to live with this thing. Why are we going to have to learn to live with this thing? Number one, asymptomatic carriers. Because there are asymptomatic carriers, we're never fully sure that we, in fact, have eradicated it. So asymptomatic carriers, that's reason number one why we'll never really be rid of this thing. Two, this virus is a zoonotic virus. It can grow in lots of different animals. Have you guys heard about this new strain from Denmark? You know what it grows in? Mink. Denmark has lots of mink farms. There's more mink than people in Denmark because of the mink industry. It's quite lucrative, which is interesting given the fact that it's completely un-PC in Europe. If you wear a mink coat in Europe, any place in Europe, you'll be shunned. The social crush against wearing animal furs is very, very strong in Europe. But it is primarily Asian markets. But there's a new strain of SARS-CoV-2 that has a mink. And now this mink strain has been passed back to humans. The virus also can infect cats and to a lesser extent dogs. So this virus will always have animal reservoirs. So will we ever be rid of things? No, because it's going to bounce back and forth between various animal species. Also, we're probably never really going to have a completely efficient vaccine or antiviral against this thing. So in 2017, can you venture a guess as to how efficient the flu vaccine was? 38%. 38%. Yes, I know the Pfizer vaccine is supposed to be 90% effective. I'll believe it when I see it. We will never have a vaccine that's completely effective for this. We'll never have antivirals that are completely effective for it because of the nature of the disease. We'll talk about it a little bit today at the seminar. So we're going to have to learn to live with this thing. So that is the unhappy truth about COVID-19. So on that somewhat grim note, let's go ahead and jump into our topic at hand. And let's have a word of prayer. Father, thank you for this cold day. Thank you for the end of this week. Thank you for the end of our labs and the students who all worked so hard, who came in on Saturdays, who came in off hours. Lord, thank you that we're almost through into the home stretch of this semester. Help us use our time wisely. Prepare us for finals. Help us to all live wisely, stay well. In Jesus' name, amen. All righty. Let's see here. So, can you please get out your handout that has all the pictures of antibodies on? So we have a summary of the antibody subtypes right here. Now, IgD. IgD is expressed on the surface of very young B cells. It's essentially a, how should I put this, driver's license for the young B cell that tells the young B cell, you're ready to go out into the reaches of the circulation and try to tackle actual antigens. So this is never secreted. It's just a driver's license, it's co-expressed in the young B cell with IgM. So this is expressed together with IgM. It's never secreted, it's only surface bound, and it serves a regulatory purpose only. IgM. When it's secreted, it's the first antibody secreted by the young B cell, secreted as a pentamer. And the book has a picture, essentially, of an IgM pentamer, and I think you have a picture on that handout of an IgM pentamer, do you? Yeah, so there's 5 of them. That means there's 10 antigen binding sites. So these things have very high, what's called avidity. Avidity. That means it doesn't necessarily bind the antigen tightly, but it can handle lots of different antigens at one time. So I played football when I was in high school. We had a kid, he was wide, he was over 300 pounds. His technique was terrible, but just by getting in the way, that guy could probably blow out 3 people. So his avidity was high, his affinity was low. So that's the thing about IgM, high avidity. Not good at opsonizing. Very, very good at fixing complement. That's what IgM excels at, is fixing complement. It's secreted as a pentamer. Now, gang, remember this. All of these antibodies, both a form that's on the surface of the B cell that's embedded in the membrane and a form that's secreted. The form that's secreted is the same as the form that's in the membrane. All this cell needs to do when it sees messenger RNA, there's a little piece at the end of the antibody that encodes for a piece that sticks it into the membrane. If it includes that piece, it's stuck in the membrane. If it takes that piece out, making the mature RNA, it'll be secreted. But all antibodies will, when it makes a secreted form of an antibody, will have that same antibody embedded in the membrane. So there's a membrane-bound form and a secreted form. Does that make sense? So it's kind of like a button telling people how you're going to vote. So your vote would be the secreted part, the button on your clothing or your lapel, whatever. That's your membrane-bound form. IgG, most abundant antibody serum. Excellent opsonizing antibody, probably the best at opsoning. Can fix complement fairly well. Not as good as IgM, but it can still fix it. There's 4 different types of IgG, 1, 2, 3, and 4. I don't want to get that right now, just for our purposes, IgG. IgA is secreted as a dimer. Secrete a dimer. And you can see the dimer up there. This is the one that is exported across mucosal surfaces. So this is the primary antibody mucosal surfaces. Good opsonizer, not a very good activator of complement. Used to be thought IgA didn't activate complement at all. Turns out it can by the lectin pathway, but it's not very efficient. So really good complement actor, pretty good complement activator, not so good complement activator. Terrible opsonizer, good opsonizer, pretty good opsonizer. IgE. IgE is secreted during allergies or during parasite infections. Good opsonizer. Certain cells. Does not act complement at all. And as you can see, IgE is very low in blood. If there's lots of IgE in your blood, there's a test for that. It's a RAST test. High IgE in your blood is an indicator you're suffering from allergies. So if you suffer from asthma or seasonal allergies, that's going to be a good indicator. And you can just order a RAST test, R-A-S-T, RAST. Are there any questions about the different types of antibodies? Again, antibodies are made by B lymphocyte. B as in Berkshire Farms, or B as in bone marrow. Those are the cells that secrete antibodies. B antibodies. Okay, now let's take a look at the life and times of the B cells. Please take this handout out. Look at this first panel here. So the B cell is born in the bone marrow and is born from the hematopoietic cell. The daughter cell becomes a lymphoid precursor, and then that lymphoid precursor, under the influence of various cytokines in the bone marrow, will bind to a stromal cell in the bone marrow. Binds to a stromal cell in the bone marrow. So let's make the stromal cell red here. That's our stromal cell. Then we'll make our B lymphocyte in, where'd my black marker go? Black marker seems to have walked. Okay, we'll do the B cell then in red. Now, this B cell is going to attach to the stromal cell. So this is stromal cell here. You're going to get specific attachments between these two cells. They both express adhesion molecules that invite these two to attach to each other. And this stromal cell is going to secrete interleukin 7. Interleukin 7. This constant secretion of interleukin 7 is essential for the continued life, survival, and maturation of this B cell precursor. So that's the first step in the B cell's life. So the B cell then is going to go through these several different stages. Listed here. Go through these with me if you would, please. 6 different stages. Starts out as a lymphoid progenitor, then it becomes an early pro-B cell, late pro-B cell, large pre-B cell, small pre-B cell, and finally an immature B cell. Those are the different stages. Let's go through each of those stages and discover what happens to the B cell. Now, the B cell has 3 antibody main genes. Remember, the antibody consists of a heavy chain, 2 heavy chains, and 2 light chains. A heavy chain and 2 light chains. The heavy chain is encoded by a gene cluster on chromosome 14. There are 2 light chain clusters. They're called kappa and lambda. Kappa, K-A-P-P-A, and lambda, L-A-M-B-D-A. Kappa is on chromosome 22. Sorry, chromosome 2. Lambda is on chromosome 22. The heavy chain's on chromosome 14. Now, the picture below, it shows you essentially the composition of one of these heavy chains. So the heavy chain contains several V gene segments. V stands for variable. There's 44 of those. It has 27 D segments. D stands for diversity. And you can see those there as well. Then there's 6 different joining or J segments. Now, why does this gene have all these different segments? It's to create diversity. Think of it this way. Say you have several different sweaters or blouses, let's say. And then with those blouses, you have several different sets of skirts or trousers or leggings or whatever. Then on top of that, you have all different types of shoes to go with each of those different types of outfits, different types of socks or other types of footgear or something like that. By mixing and matching each of these, you could develop a multitude of different outfits. So your number of outfits is far greater than your number of individual tops, individual trousers, individual shoes. It's the very combinations of all those that'll give you the number of outfits. That's how these antibody genes create such huge diversity. It's going to pick one of those V chains, one of those V segments, one of those D segments, one of those J segments, and it's going to join them together. And that's what's going to give you heavy chain. And with the light chain, you have a V and a J segment. There's no D segment on the light chain. You can do the same thing for the light chain, and that's going to give you enormous potential for diversity. That's why antibodies are able to handle so many different types of antigens, because of this huge amount of diversity. All right, is that part clear before I move on? Yes? I'm particularly proud of the wardrobe example. I have to credit my middle daughter for that one because she's such a close horse. Okay, now, when the B cell expresses a gene called the recombinase for immunoglobulin genes 1 and recombinase for immunoglobulin genes 2, RAG1 and RAG2. That is what's going to help you take those genes and properly rearrange them. So when it expresses that particular protein, it is now an early pro-B cell, an early pro-B cell. RAG1 and RAG2 will essentially pick at random one D and one J segment, and it joins them together. How does it do that? All the DNA intervening, RAG1 and 2 finds it, loops it out, cuts it out, and then just takes those two and glues them together. So it does the ultimate cut and paste thing. My wife was making something yesterday. She works at elementary. She was making something for the kids, and it was too big. So she goes, snip, and then she glued them together. And I said, boy, that's just like antibody rearrangement. She didn't see it that way, but I did. So when it rearranges D and J so that it has a proper D and J segment glued together, it now becomes a late pro-B cell, a late pro-B cell. You can see that essentially on your handout where it says late pro-B cell, the DJ rearrangement has occurred. It begins in the early pro-B cell stage. When it's completed, you have a nice late B cell. Then this same recombinase with additional enzymes will now pick one of the V regions and it'll do the same thing. Takes the intervening DNA, clips it out, glues it together. So you get a V a D and a J segment that have all been glued together. And when you do that, you now have a large pre-B cell, a large pre-B cell. Now you need to see if this heavy chain that you've made is functional. How do you do that? You express it on the surface and you put together with it a surrogate light chain, a surrogate light chain. So it makes a little surrogate light chain, and if that surrogate light chain and the presumptive heavy chain form an antibody-like molecule on the surface, it sends a signal inside of the B cell, and now the B cell starts to proliferate. It starts to rapidly proliferate, and each daughter cell is a small pre-B cell, a small pre-B cell. Now, the small pre-B cell, all of the small pre-B cells have the same heavy chain, but each one of them will have a different light chain because the light chain has not been arranged yet. Do you see how this is essentially rigged to give you maximum diversity? Really kind of cool when you think about it. Now you will rearrange the light chain. You rearrange the kappa chain first. If you don't get successful rearrangement of the kappa chain, then you move to the light chain. Now you might wonder what happens if the heavy chain is not rearranged successfully. There's two copies of the heavy chain. If the first chain doesn't get rearranged successfully, then it goes back and does the other allele, the other copy of it. If that fails, then the cell dies. And there you are. So we essentially now have this rearrangement of the kappa. If that works, then we don't rearrange the lambda chain, but if it doesn't, then we rearrange the lambda chain. And then we try to express them on the surface. If we express them successfully on the surface, we have an immature B cell, an immature B cell, and this immature B cell is now released into primary lymphoid organs. Primary lymphoid organs. These primary lymphoid organs include the spleen, lymph nodes, and tissues like the big one would be the spleen. Now, in primary lymph node organs, you have a gene that's called the autoimmune regulatory gene, or autoimmune regulatory enhancer, actually. This essentially will cause primary lymphoid organs to express a whole gamut of self-antigens. Now, why on earth would it do that? Here's why. So think of this kind of like, any of you the Epcot Center? Epcot Center is kind of a microcosm of the whole world. There's a representation essentially of things around the world. That's what primary lymphoid organs are like for this developing B cell. It's like an immune Epcot Center. So what these cells do is they go and they sample antigens from all over your body. If they cross-react to any of those self-antigens, the cell gets offed. It gets killed. Why? You don't want an immune cell that interacts with their own antigen. And people who have mutations in this particular gene will suffer essentially from a condition that prevents them from making anything except IgM. Because these cells will never go on and mature into anything bigger than that. All right, so the cell then, when it passes essentially through these primary lymphoid organs and it doesn't cross-react, then it will express IgD on its surface. And ID, again, is the driver's license that tells the body this cell's okay. This one can drive. It's been screened. It's okay. You're fine. You can go out in the world and drive to go see your friends in Indiana or whatever, or something like that. And now the immature B cell will circulate or will take residence inside lymph nodes. And if it reacts with an antigen that is specific for that surface IgM, it will then go on and mature and be activated. And when the cell becomes a fully mature antibody-expressing cell that doesn't divide anymore, it's called a plasma cell. A plasma cell. Plasma cells don't divide. Immature B cells, when activated, they divide like mad. Do you remember from pathophys tumors of plasma cells? Remember what those are called? Anybody? I know you hate it when I do this to you, but I'm trying to stretch your memories because you'll have to stretch them when you take the NCLEX. Multiple myeloma. Multiple myeloma. That's a tumor of plasma cells. Tumor of plasma cells. All right, so we have the rearrangement of the heavy chain on that page, and then on the second page, you see the rearrangement of the light chain, and that's all essentially tied in with the life cycle, essentially, of the B cell. Are there any questions about B cell life? No? I know it's a lot, and I know it's kind of esoteric information, but think about it, ruminate on it. If you have any questions, go ahead and email me or ask me Monday or something like that. Okay? Well, good. All right, everyone, take a nice deep breath. Let's now move on to what happens when you activate a B cell. And that is the subject of this picture right here on page 2. So page 2, the lower figure. Now, your B cell, it expresses IgM, it expresses IgD on the surface. It is now mature, but it's still naive. Naive. What does that mean, naive? Well, it means that it has not yet experienced antigen, and it has not yet become activated. Naive. So the B cell now starts to explore the body's lymphatic system. They travel from lymph node to lymph node in search of antigen. When the B cell encounters antigen that fits its IgM molecule, there's a signal sent to the inside of the cell. That signal is now going to cause the cell to become activated. And when it gets activated, it will start to divide. That activation, however, is going to do some other things. It is going to cause the B cell to make cytokines, and the cytokines that it makes will include, I'm going to have to erase this. So interleukin 1, so activated B cell. So it divides, makes interleukin 1, interleukin 2, interleukin 6, and then tumor necrosis factor alpha. It also upregulates molecules that cause the cell to survive. And then the cell, once it's activated, will also undergo isotype switching. Now, the isotype switching means that it goes from expressing IgM to some other antibody like IgG or IgA or IgE. So IgM antibodies, again, are the first antibodies that the B cell makes. So if we take a look essentially at our handout here, you could see that the constant regions determine what type of antibody it is. So we have that section there that says mu, that's the Greek letter mu. If you express the mu gene means that you have a mu constant region, that means you're an IgM. If you have that delta region there, that means you're an IgD. Then you have all these other potential constant regions, gammas. Well, if it's a gamma, then you're an IgG. If it's an alpha, then you're an IgA. If it's an epsilon, then IgE. With isotype switching, these constant regions essentially get excised by the machinery in the cell, and you can see that excised DNA is going to be degraded, and you essentially take the V, D, J, and you put them right next to the particular type of constant region that you're going to express. So these antibodies here, G, A, E, IgG, IgA, IgE, those are secondary antibodies. Secondary antibodies. Now, something else about the production of secondary antibodies. This process whereby you excise all that DNA is extremely error prone. So when you undergo isotype switching, the antibody that you make now will be made from DNA that has mistakes in it. Some of these antibodies won't bind the antigen very strongly. Those cells will not be activated by the antigen. They will undergo death because they fail to be activated. Other cells, however, will make antibodies that bind the antigen even more tightly. even more tightly. More tightly you bind the antigen, the more robustly the antigen activates the cell, and the more greatly your cell proliferation is evaded. Therefore, the immune response selects for those cells that bind, whose antibodies bind the antigen the most, the tightest. This is called affinity maturation. Affinity maturation. Because of the air-prone nature of isotype switching. Consequently, these secondary antibodies, IgG, IgA, IgE, they will all bind the antigen much more tightly than the original IgM. They will always bind the antigen much more tightly than will the original IgM. Because of affinity maturation, that is a consequence of this air-prone isotype switching. Okay. Oh, there we are. All right, if there are no questions on that, let me just pause here, see if there's any questions. I think, given the shell-shocked looks I'm getting from a lot of you, why don't we take a break early? That way you guys can rest your neurons a bit, and then we'll come in and talk about T cells. Because T cells go through a process kind of sort of like this, but slightly different. So let's take 5, take our break, and then we're going to go to this page there, then the T cells and their developmental process. Okay, it's time to resume. Now, if you look at page, well, I guess it'd be 2. Well, it's the second page, but it's a two-sided handout, 1, 2, 3. It'd be page 3 of your handout. It looks like this. This is the T cell receptor. So just as antibodies have a receptor, B cells have an antibody on their surface, that is their B cell antigen receptor. T cells have an antigen receptor. Antibodies, of course, can bind 2 copies of the antigen, whereas T cells can only bind 1 copy of it. So this particular receptor, as you can see, also has a constant region, also has a variable region. It consists of 2 chains, a cell alpha and beta chain. And this also sits in the membrane. The protein, however, is never secreted. It's never secreted. T cells secrete a lot of things. Their antigen receptor is not one of them. So this is confined to the cell membrane of T cells. Please remember that. Now, how do we get these particular arrangements? So if you turn the page, we see here, once again, we have lots of V regions, and then we have lots of J regions. We don't have any D regions for these 2 genes. And it's similar for both the alpha and the beta genes. All right, so by essentially mixing and matching V regions with J regions, we can get a particular V to J region that are joined together. By joining them together, we then also put them together with a constant region. We secrete them to the surface. We have then the proper T cell receptor. You can see that for the alpha chain, there's between 70 to 80 of the variable regions. There's something like 61 different J regions. The T cell will select 1 variable region, 1 joining region, and glue them together. So these cells, they rearrange their DNA. Same thing with the beta chain. there are up to 52 different V regions. That's 6 different regions. There's a constant region, a diversity region, but just one, and then 7 other types of joint regions with a second constant region. So we can generate a good deal of diversity by mix and matching, again, just like your wardrobe. Different tops, different trousers, different shoes. By mixing and matching those together, you get different outfits. At the total number of outfits are far greater than the actual number of shirts and shoes and trousers and so on and so forth. Now, the T cell is born in the bone marrow, just like B cells, but unlike the B cell, which expresses adhesion molecules to stick to stromal cells, T cells do not. Instead, T cells will move to the thymus. So the thymus is this organ that sits over the top of the heart. This is the university that will school T cells before they can be unleashed upon the body to do their job. So the thymus is T cell university. And that's why T cells are called T cells. It's T cell after thymus. B cells are called B cells after bone marrow. They're more complicated than that, but you get the point. Now, when the T cell travels to the thymus, the thymus has an outer cortex and an inner medulla. Outer cortex and an inner medulla. If the T cell does not properly rearrange its genes for the receptor, it's done for. The cells within the thymus will detect that this cell doesn't have a proper antigen receptor, and it'll persuade the cell, induce the cell to undergo programmed cell death. All right, so the first thing that the cell does is that it rearranges its alpha chain. No, sorry, beta chain. Rearranges the beta chain first. Now, there are 2 molecules that are going to designate which kind of T cell you have, and it will express these at different times in the thymus. One of these proteins is called CD4. And if you express CD4, you are a helper T cell. The other is CD8. If you express CD8, you are a T cytotoxic cell. CD4 cells play a role in assisting other cells of the immune system to do their job. CD8 cells kill infected cells. So these cells are the executioners. These cells are the enablers. The executioners and the enablers. Now, early stage, the T cell expresses neither CD4 nor CD8. Instead, it is rearranging its beta chain for the T cell receptor. So these cells are called DN cells, double negative. They are negative for CD4 and they're negative for CD8. Negative for CD4, also negative for CD8. So once it starts to rearrange its genes, if it makes a beta chain that is successful, you can see there's one of these cells that's called a DN3 cell. A DN3 cell. The DN3 cell properly takes a D segment and puts it next to a J segment. So you have proper D to J rearrangement. That's a DN3 cell, still double negative. Then it's going to rearrange that V region. It's going to select a proper V region. If you get a proper VDJ rearrangement, it's a DN4 cell. A DN4 cell. The cells of the thymus, the stroma of the thymus, are going to express interleukin 7 and also interleukin 2. These are growth factors for T cells. These are essential for T cell maturation and survival. So you rearrange your beta chain, DN4. Now you are properly rearrange your beta chain essentially. Now you're going to test out the beta chain, and you're going to express it with a surrogate alpha chain. With a surrogate alpha chain. If that is successful, you get a proper beta chain with the surrogate alpha chain. That sends a signal to the inside of the developing T cell. And now the T cell will express both CD4 and CD8 on its surface. Now it becomes a double positive cell, or a DP cell. A DP cell. It expresses both CD4 and CD8. At this time, the alpha chain now undergoes rearrangement. If the alpha chain undergoes successful rearrangement, it will then express its T cell receptor with the CD4 and CD8 genes. And now it'll start to sample antigens from all around the thymus. And now the cell, and you can see this, the cell will go from the cortex to the inner medulla of the thymus, as you can see on the following page. If at any time that T cell receptor interacts strongly with a self-antigen, the T cell is killed. This is a very selective filtration / screening process. By any self-reactive T cells are essentially given the signal to off themselves. Now, the T cell as it interacts with various antigens, those antigens are presented to them on a platter. What's the platter by which they are presented? We already talked about this platter. It's those major histocompatibility proteins. So these antigens that are being presented to the T cell, they're presented on a platter of these MHC proteins. There's class 1, there's class 2 MHC proteins. These have different structures, and the way they bind their antigens is very different. We'll get to that in just a bit. But if the T cell finds a foreign antigen that it interacts with, and that antigen is presented by a class 1 MHC, then this double positive cell will downregulate CD4, and it'll upregulate CD8, and it becomes a CD8 T cell. It becomes a T cytotoxic cell. If, however, that double positive cell finds that its T cell interacts with a foreign antigen presented by a class 2 MHC, then it will downregulate CD8 and upregulate CD4. And now it becomes a helper T cell. So the antigens presented by these cells in the medulla of the thymus are both self and non-self antigens. If it interacts with a self antigen, it gets killed. If it interacts with a foreign antigen, depending upon which MHC that presents the antigen to it, it will become either a CD4 positive T helper cell or a CD8 positive T cytotoxic cell. Once the cell has a properly functional T cell receptor complex with either CD4 or CD8, the T cell will be released to the general circulation. Only after it completes this very, very intense screening process will it be released, because nothing hurts more than a T cell that interacts with self tissues. And on the top, essentially, this part of your handout, you can see the two different types of T cells. So you have one T cell that has as its co-receptor CD4. CD4 T cells will recognize antigen presented to it by class 2 major histocompatibility proteins. On the other, left-hand side, you can see a T cell receptor complex with CD8. CD8. These cells will recognize antigen presented to it by this major histocompatibility class 1 proteins. And they will be T cytotoxic cells. And since we're on the subject, let's talk about antigen presentation. That's on the back page of your handout. So let's say, nurse, you're going through the hospital floor, you come into a room, you look down at your iPad or whatever you might have, you look for this patient's ID, it's not in there. Patient doesn't have a wristband either. You don't know where the patient came from. Can you treat? No, you don't have a diagnosis. They've not been checked in, not in the system. For all they know, they could be an assassin who's there to take out one of the doctors. So what do? You call security. Same thing with the immune system. The immune system does not recognize something as properly foreign unless it's presented to it on a particular presentation form. That presentation platform are these MHC proteins, type 1 and type 2. Here's how they do it. Class 1 MHC proteins. MHC present proteins, carbohydrates like that. You have some sort of internal virus, infective agent, whatever. You have something that has gotten into the cell. Whatever that thing is, has gotten into the cell, it's degraded by the proteasome in the cell. The proteasome acts like the clean garbage disposal of the cell. Chops up some of the proteins so that you have amino acids that you can then recycle. Now, sometimes it chops it up and you get little pieces of the protein, little polypeptide chains. You have a transport protein in the endoplasmic reticulum called TAP. TAP. Transporter of antigenic peptides. The transporter of antigenic peptides forms a complex, TAP1, TAP2. It sucks those into the ER lumen. In the ER lumen, it will bind the MHC class 1 proteins because that's where MHC class 1 proteins are made, in the ER. So since they're made in the ER, they will bind those MHC class 1 proteins in the ER. Then that protein will be packaged into a vesicle, will then be sent to the cell membrane. So these are antigens that find their way inside the cell. Things like viral antigens, viral proteins, viral nucleic acids, things like that. What about class 2? Class 2s. Class 2s begin their life again in the ER, but then they are sorted in endosomal vesicles. Now, these things that will be presented by MHC class 2s are things that come from the outside of the cell. The cell brings them in to the cell via phagocytosis. And so you have a phagosome. That phagosome will break down these proteins or carbohydrates into smaller bits, and they merge with the endosome. In that endosome, you have class 2 molecules. They will bind these proteins. Then those MHC class 2 proteins will be sorted from that vesicle and to the membrane. And there the class 2s will hold up that antigen as if to say, here it is, recognize this, that's foreign, so on and so forth. So if a class 1 holds it up, it's going to be recognized by a CD8 expressing T cell, because CD expressing T cells recognize antigen presented by MHC class 1. So that'll be viral proteins, intracellular parasites, things like that. And if they recognize what T cytotoxic cells do, they'll destroy that cell so that the viral replication process is cut short. The intracellular parasite is now released into your body and can be attacked by the immune system and so on and so forth, instead of hiding inside your cells. The class 2 MHCs, these are things that come from the outside of the cell. They will be recognized by T cells that express CD4, CD4, and these will be T helper cells, and they will express molecules that will help B cells and so on and so forth. All right. Anything in all of that you want me to go over again? Any questions you have? Anything that's not as clear as you'd like it to be? Go ahead and ask questions now. No questions? If you don't understand all of it right away, don't feel badly. It's complicated. You may have to read it over 2 or 3 times, and then it'll start to come. It's kind of like me in John chapter 7. I just can't remember it very well. I have to go over it multiple times. So if you don't understand this right away, don't feel badly at all. It's kind of the way it goes. All right, let's talk about activation of these cells. Now, what if cells happen to bind antigen and present it mistakenly? Will the cell still react to it? The answer is probably not, because there is a second check on the activation of the immune system. What is that check? You require a 2-signal activation system in order for the T cell to actually get activated. Let me illustrate here on the board. Look what I found. It was hiding. All right, so let's say you have a dendritic cell. Okay, it's ugly, but you get the point. It's in a dendritic cell. Dendritic cells are antigen-presenting cells. So let's say this dendritic cell has on its surface an MHC class II. And this is presenting some sort of antigen. What kind of T cell is going to recognize this? Will be CD4 or CD8? 4, CD4. So its receptor and then it's going to have a CD8 molecule, and that CD8 molecule is going to form a proper interaction. So this is the T cell receptor, and this is CD8, and this is a helper T cell. You require actually a second signal. The second signal is going to guarantee that this cell now knows that this antigen presentation by the proper cell What if that patient walk into the room, patient has no wristband in the system? What if all of a sudden the janitor comes up and says, oh, it's okay, I checked her in? You gonna buy that? No, there's a procedure for check-in. Right? That procedure has to be followed religiously, right? So you have a second signal, and the signal goes like this. There is another protein on the surface of the T cell, and it's called CD28. CD28 has to bind to a co-stimulatory protein on the surface of the antigen presenting cell that's called B7. This two-signal system, this now tells the T cell together, both of those to say, number one, this is a credible antigen presented by a credible cell, I now be activated. So you need a two-system activation system in order for the cell to properly react to antigen. So you need the T cell receptor binding to a properly presented antigen. We call that an antigen presented in the proper context with a second signal, CD28 and B7. Okay. Once that happens, the T cell is now going to rev up. First thing it does is it makes interleukin-2. Interleukin-2 is a hugely important growth factor for T and B cells. When it makes interleukin-2, it also makes the receptor for interleukin-2. So interleukin-2 does this really cute trick of activating the very cell that made it. This is called autocrine signaling. Autocrine signaling. Autocrine signaling. The cell now starts to divide. Starts to divide, and it will now start to secrete, depending upon what specific type of T helper cell it is. But one of the proteins that T helper cells can secrete is interferon gamma. Interferon gamma. What interferon gamma do? Interferon gamma is going to stimulate macrophages so now they're much better at intracellular killing than they would be normally. So these cells, if you have, for example, a tuberculosis infection, the macrophages normally will pick up the Mycobacterium tuberculosis. They don't do a very good job of killing it. So what they do instead, they just encircle it and cut it off from the air. If, however, T helper cells make interferon gamma, now these macrophages in the lung, when they phagocytize Mycobacterium tuberculosis, can kill it. Now they can kill it. The other thing interleukin-2 does is it also is going to stimulate the proliferation of B cells. It will also stimulate proliferation of natural killer cells and also T cytotoxic cells too. So think of interleukin-2 as a generalized lymphocyte factor, generalized lymphocyte growth factor. Now, type of antibody that the B cell is going to make is going to depend upon what type of T cell is around when it gets activated. So incidentally, I don't know if your book uses this term. I'm not sure. This collection of receptors whereby the T cell interacts with the antigen presenting cell, this is called the immune synapse. Don't remember if the book used the term or not, but that's the term it's usually used, the immune synapse. Okay, now what determines what type of secondary antibody that it makes? It's going to depend upon the cytokines secreted by the specific antigen presenting cell and T cell that interact with specific. It turns out different antigens elicit different cytokine responses from these cells. So, for example, if you have the APC cell churning out interleukin-4 and interleukin-5, they're going to induce B cells to isotype switch to make IgE. IgE. If it tends to secrete more, let's use a different color here. More interleukin-1 and interleukin-6, it's going to tend to cause the B cells to secrete IgG. And, oh, there it is. And if it secretes transforming growth factor beta and interleukin-5, then that cell will secrete IgA. So the isotype switching is going to depend upon which cytokines the naive cell is exposed to. These will be expressed by the APC and by the particular T cell that is activated by that particular antigen. Okay, so is going to go on. It's going to help macrophages become more stimulated. Others that'll make It'll make interleukin 17. This is going to recruit neutrophils. To the site. Some immune responses are characterized by lots and lots of neutrophils. That's due to the fact that you get expression of interleukin 17. And then there are others that are actually going to stimulate things like mast cells and basophils and things like that. And that's interleukin 13. So that's going to be mast cells, basophils, and stuff like that if you have a parasite infection or something like that. So these cytokines made by this activated T helper cell, depending upon which particular type of T helper cell it ends up being, are going to activate different arms of the immune response. That's the case for the T helper cell. What about our T cytotoxic cell, our T toxic cell? T cytotoxic cells, like T helper cells, also require the 2-signal. The 2-signal system in order to be activated. And activated if they go on to recognize antigen presented by MHC class 1 in an infected cell. They do not need a second signal. They need a second signal to be activated by the APC presenting it. Once active, however, they don't need a second signal in order to kill this cell. They find cells presenting the antigen with MHC1, those cells will be killed. The cytotoxic cell will make the perforin, and they will make granzyme B. That will cause the infected cell to die very effectively, in fact. Now, what about our other friend, the natural killer cell? Natural killer cells, their mechanism is not very specific, as it turns out. So, natural killer cells don't respond to presented antigen. Instead, natural killer cells, so let's say, cell number 1, cell number 2, different color, cell number 3. And then we'll do a natural killer cell here in green. Okay, what's this looking for? There are on the surfaces of most of our cells, first of all, major histocompatibility class 1. And then there are other ligands that tell the natural killer cell, don't kill me. So this one will come up here. It sees this. It sees these other ligands, and it leaves it alone. It then toddles over here. Is the MHC1? May have an antigen in it, doesn't care. Has some other sort of maybe don't kill me signals here, it's fine. Comes over here, what does it see? No MHC1. When it sees no MH1, that's a signal that the cell is normal. And once it does that, it will bind to it very tightly. It will secrete perforin, granzyme B, and this cell will be killed. So our cells have signals that say, kill me, to the natural killer cell. If those signals disappear, the natural killer cell will then come up to you and go, no, something is hinky here, and it will kill the cell. Now, what do you have a cell that is virally infected and is expressing lots of viral proteins on its surface? And then this gets coated with antibodies. So on and so forth. Natural killer cells have receptors for the Fc portions of antibodies. If it sees a cell coated with antibodies, it says to itself, no, not cool. So it will then bind to these antibodies and it will initiate a process called antibody dependent cell cytotoxicity. And this cell will be killed. Like that. So natural killers do not respond to presented antigen. Instead, they respond to cells coated antibody or to cells that don't have the proper array of receptors on their surfaces. That's why natural killer cells are so important in staving off cancers, because it turns out tumors probably arise at a certain rate in your body, and natural killer cells are busy constantly getting rid of them. And it's starting to weaken as you get older. That's why cancer tends to be more a disease of the aged. Okay, I see it's 9:15. 9:15. We'll finish this on Monday, and then we'll get into the diseases. So hope to see you at 10 o'clock. And your lab books are here. If you want to turn in your lab book, let me know. If you've picked yours up, let's see. Okay, yes, Karis, Emily. Okay, if you want to hand them in, let's start a new pile. There. Okay, have a good weekend, gang. Good gravy. All right, there we go.